From Microbe TV, this is TWiEVO, This Week in Evolution, episode 17, recorded on March 6, 2017. This episode of TWiEVO is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first purchase with free shipping by going to blueapron.com/twee. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today right here in New York City, Nels Elde.
Great to be with you in vivo again, Vincent. Yeah.
Nels is here in my office.
I'm here. I was just jogging along the banks of the Hudson River.
You were.
And thought, I should swing up to headquarters.
Well, anytime anyone's in New York, they should come by to the
that's right.
studio here.
Agreed.
And so good to have you here.
Good to be here. Beautiful, crisp, sunny, late winter day.
It is nice out.
Got some fresh air. And
chilly, though.
A little chilly.
This morning
crisp.
it was freezing, yeah.
Yeah, but that's okay. You just kind of get moving and huddle with the New York crowds, and you can get through the day.
We have a guest here in studio who is no stranger to me because he is his office is next door to me. He is actually, I should say first, he is a Howard Hughes Medical Institute investigator, and he's also a Higgins Professor of Biochemistry here at Columbia University. Steve Goff, welcome back.
It's good to be here.
Well, first time on TWiEVO.
First time on TWiEVO.
But he's
great
been on
honor.
TWiV twice or 3 times or something like that.
Mm-hmm.
Yep. And as you say, I'm next door. I had to come all of 10 feet to get here,
right.
So it was tough. I didn't have to jog, but
you're a Higgins professor. So am I.
Like you, yes.
Yeah, Henry Higgins. You know that story?
No, I don't. That's a long one.
That's from My Fair Lady, I think.
Oh, I see. Okay.
Maybe I don't know who Higgins was.
No, Eugene Higgins, not Henry.
Oh, it's Eugene Higgins.
Yes.
All right. Well, we're in good company.
I agree.
Eric Kandel is a Higgins.
Half the people at Columbia, at least.
Right. Well, then we're in good company. So, Nels, what brings you to New York City?
Yeah, so it wasn't just to jog along the banks of the Hudson River, as nice as that is. So I got into town Thursday night in time for a symposium and workshop this weekend on social behavior in genome evolution. A little outside of my comfort zone, which is what kind of got me interested and excited to do this. So this was a small meeting organized by a fellow Dustin Rubenstein, who's in biology at Columbia, in the Morningside campus, a little bit south from you fellows. He studies starling birds in Kenya, and also more recently these really crazy eusocial shrimp species.
What does eusocial mean?
Very social.
They're very,
like
the opposite of asocial.
Correct, yeah.
Okay.
Yeah. So these are not like Scandinavian
They're
shrimp.
the opposite.
These are they're not shy.
Opposite of scientists.
Correct. That's right. Yeah. And then the meeting was co-organized by a fellow called Hans Hoffman. He's at UT Austin, neuroscientist and also an evolutionary biologist. And so the idea of this symposium was to bring together folks who work on social evolution, social behavior, the biology of this, and then also people are thinking about this sort of a genomic level, and even just genome, sort of thinkers and evolutionary thinkers. So it's kind of a mix of folks. So, and I have to say, a lot of the work on social behavior is being done in insects. All the interesting biology that we've collided into from time to time seems to happen in insects. Yeah. And so a few of our past TWiEVO guests were on hand. Corey Moreau, for example, from the Field Museum, who studies ant behavior and these symbiotic associations with bacteria. A couple of people studying honeybees as well. And so all of this range of and other ant species. And so all this range of complex behaviors that can emerge from these insect societies. And then what you would love to do is to sort of get a genetic handle on maybe how some of these more complex behaviors emerge. This really interesting and also in some ways I would say elusive field of behavioral genetics. So another TWiEVO guest, Hopi Hoekstra, was here. She does this incredibly beautiful work on many social behaviors in mice from the Peromyscus genus. And so she was talking about some recent work from a postdoc of hers, a fellow named Andres, who will be opening his group actually here at Columbia this summer, looking at parental care and a genetic handle or two they have on that using forward genetics. And so we had a range of people really thinking about complex behavior, people using sort of bridging, using genetic approaches to get at this, and then folks thinking about how genomes evolve and whether there's a way to sort of find some middle ground. Really interesting woman, Lucia Carbone from Oregon, who is one of the world's experts in Gibbon genome evolution. Gibbons turn out to be really wild when it comes to their genetics. It's as if a bomb went off on their chromosomes, all kinds of inversions, rearrangements. Yeah. And she's traced this in part to a set of really unusual retrotransposable elements, these so-called lava elements that are sort of a Frankenstein of several different flavors of retrotransposons that have come together and then have sort of expanded or proliferated in these genomes. And there's this idea that there's some connection between that and these wild genetics that have ensued among the gibbons.
So they're very polymorphic. The individuals are just very different from each other in their endogenous
That's right,
repertoire.
Yeah. And there's been sort of this burst of species diversity. So there's something on the order of a dozen or almost maybe 20
subspecies.
Exactly. And those sort of borders are all blurred. The definition of what's a hybrid or what are fertile sort of species boundaries there. And this is reflected. So if you compare, a close any of the other hominoids, so humans, gibbon or bonobos, chimpanzees, gorillas, orangs, the arrangement and the structures of our close relatives, those genomes are pretty well behaved. However, the gibbons, it's just completely scrambled.
Yeah. I was going to ask you, is it different from the other non-human primates? But it is, huh?
Quite different.
We don't know why, though, right?
We don't. So there's that kind of intriguing hook that these Frankenstein lava elements, retrotransposons, could have something to do with it. But that's sort of where the active...
Are they gibbon-specific, those lavas?
They are indeed, yeah. So they're mixes of lines and alus and other hybrid elements sort of all packed together and then autonomously propagating through genomes. So, yeah.
Did you know about this meeting?
No.
I didn't either.
No.
Yeah, it was kind of interesting, so I think...
Yeah, it would have been fun.
Yeah. And so it was open to the public on Friday, and then Saturday, Sunday, the folks who came to speak all sat down, and we were working on a review paper.
Sorry to hear that.
Yeah. Yeah, it was an interesting...
No, it's a good thing to do.
Good thing,
right?
Yeah.
It's work, but it's a good thing.
Where's it going to go?
So there's a journal called TREE, and I'm forgetting what that's an acronym for, but that sort of handles a lot of work sort of in this area. I, of course, was trying to bring in a sort of microbiologist's or virologist's view to the conversation with this idea that viruses, for instance, as they've been deployed, can in some occasions manipulate host behavior in really interesting ways. And so there's one example that was recently uncovered by some folks, this Afla virus, which is a very small RNA virus, infects insects. It's actually co-injected upon infection or infection by these parasitic wasps that lay eggs in ladybugs. And then the virus has these neurotropic properties and is proposed to go to the ladybug brain, change the ladybug into a zombie, and then the bug protects the developing wasp larva from predators. And then where it gets really wild is once the new wasp has developed and flown away, that virus infection can resolve, and the ladybug can go back to its sort of regular business. And so there's all this crazy natural selection. And the idea being that then these viruses might give us a clue or a window into what are the sort of genetic footholds for how you gain access to controlling behavior. And there's some other examples like that. So.
That sounds like a perfect probe for things that are otherwise really hard to access.
Yeah, the viruses have sort of figured it out.
As always, viruses tell you what to do next.
That's right.
Is this Afla virus story new? relatively new?
Relatively new. So it's in the Royal Proceedings Journal, Part B. There's a local now, actually, a woman, see if I'll remember her name, Nolwenn Delhi. She just opened her lab at Stony Brook, working on host-parasite interactions. And she did some of the electron microscopy, both in the parasitic wasps to observe these Afla virus particles, and then on the ladybug brains to build the case that these things are actually transmitted across as part of this developmental cycle.
How do you spell that?
Last name Dheill.
Dheill.
Yep. First name.
And the virus is Afla or Ifla?
AFLO, I think.
AFLA or AFLO? Yeah.
I'm not sure.
One or the other. We'll get a link up to this. It's a really cool paper. And then I'll return to this a bit later in our pick of the week section. I'll follow up on a few more details.
Delhi N is the first name N? Okay.
Correct. Yep.
It's interesting because I was at was teaching in Jonathan Weiner's class this week. He's a science writer. Of course, he's well known for Beak of the Finch. Got a Pulitzer Prize with that. And he's really interested in viruses that alter behavior, right? He wanted to talk about that a lot.
Oh, interesting. Yeah.
It's not what I do. I'm sorry.
Yeah.
This is a good one. He would love this one.
That's a great one. And then there's another classic case. This that was observed more than 100 years ago. Wipfelkrankheit. So these are German scientists who noticed that caterpillars infected by baculoviruses.
Yeah, that's a good one.
Climb trees. And so the genetic basis of this has been proposed recently, a gene EGT, which is, I'll probably screw this up, ectodisone steroid UDP glycal transferase gene. And so hopefully in my sort of word soup there, you got the word steroid, which is, of course, this family of hormones. And then this gene encodes a protein that modifies the hormone in a way that's been proposed that then sort of provokes the caterpillars to start climbing trees once they're infected and this is that gene is expressed. And the best evidence for this is if you knock out the gene and then look at caterpillar locomotion at a certain time of the day,
right.
It's much less. And then if you complement those viruses with that gene, you replace the gene back in, now they are sort of more agitated and do more climbing.
And that's selected because it helps spread, presumably, when they're higher in the tree.
That's the
argument.
That's the argument, that as the viruses are replicating, and then actually they sort of, there's so many of them, they burst out that they broadcast more broadly for the caterpillars below to then consuming leaves will pick up the virus and the infection cycle continues.
Do you know any examples of human behavior being altered by viruses?
Yeah, great question. So, I think we're still a little bit in the weeds when it comes to humans on this case, but vertebrates, there's a famous case of toxoplasma infections of mice that then are proposed to make the mice less fearful of predatory cats. And in fact, there's the connection is in the odor of urine, in that in fact the mice actually are attracted to this instead of repulsed by it, and then they get eaten by the cat, which is an important part of the toxoplasma replication cycle, and then you start to continue that pattern as well. So then so it's a little bit out there, but the crazy cat lady phenotype is there a human? Because we humans themselves some of us around this table might have some toxoplasma infections in our brains, and at least I for one own 2 cats, so I might be, I don't know what's going on with that, but I would say the jury is still out in human cases.
Yeah.
I did sort of raise this idea at the end of my talk that, if we were looking for this, we might try to think about what are sort of behaviors that would actually promote the replication of viruses. And this could be sort of irrational or illogical things that humans might do. And so the one example I held up sort of tongue-in-cheek was the idea of the anti-vaccination campaign. And is this the result of a brain infection by a virus that has tried to propagate?
Yeah,
there was actually, I think an Ig Nobel Prize was awarded for this idea a few years ago. So anyway.
Well, there's a simple example. Respiratory viruses provoke a cough.
Yeah, absolutely.
And that spreads them. Now, it's really a host response, but it probably has been selected to transmit viruses, right? Because
That's right.
The virus gets in, it's irritating, respiratory tract, you cough, you sneeze, boom, transmission.
Yep.
I mean, so
or think about the rabies virus and what this can do to things like raccoons, for example.
certainly animals, they get crazy.
So they start biting and spreading that way. Yeah.
But people don't do that, right? People we hospitalize people, so we intervene and maybe interfere in whatever behavioral changes would have happened. So, but I think they can be subtle. They, like coughing or other things. Maybe not washing your hands is a virus-induced activity.
Yeah. Or podcasting about viruses.
Yeah, that's true. That's true. All right. That sounds like a great meeting. I wish I had known about it.
Oh, sorry. I
mean, you told me I should have put two together, but Schermerhorn is where I teach my virology course.
Oh, is that right?
Yeah, that's the floor below, 501.
Oh, sure, in the auditorium there.
Yeah.
Yep.
614, is that a small, is it a big room or a small room?
It's a medium-sized room. I think that we had about 100, 120 folks in there on Friday. Good energy in the room, great questions, and this diverse slate of talks.
Did you meet Stuart Firestein there? Didn't
see him, but I should say I just finished teaching a class with the first-year grad students at the University of Utah, and what we did was we gave them a copy of Stuart's book, Failure, Why Science is So Successful, to read over the holidays. And then we discussed it on the first day, and in this kind of some of the ideas that Stuart held up in the book, we continued to sort of use as a thread throughout the class.
Yeah.
With the idea that we're trying to get sort of beyond sort of the textbook views of science and into the real, kind of the meat of it as the grad students are starting to join labs and think about interesting and creative thesis projects that are different than sort of the old didactic lecture style of learning.
Yeah.
And they have to learn that failure is acceptable and
That's right.
even desirable.
Exactly.
At least to be expected, for sure.
Right. Yep. To embrace and celebrate it in many cases as a way
And I think we're now all entering an era where we're worried that failure will not be accepted, and that would be frightening.
100% agree.
Yeah.
It's hard to get people to accept failure, right? But that book helps because they say, oh, as a scientist, he wrote this, he believes this. Yeah, maybe that helps.
I think it's a good way to kind of shake the tree a little bit and sort of maybe recalibrate on how to think about approaching a thesis project or a PhD. And the earlier we can do that, in some cases, the better, because we're sort of primed for failure in a positive way.
Right.
Yeah.
I remember having an argument at a meeting where I thought it was really important to get people to admit that they didn't know something. Yeah. And that was perceived in a lot of scientific settings as not acceptable, that you should always claim that admitting ignorance is a sign of weakness. And I argued, no. It's very important to admit you don't know something.
Yeah, that's right, Steve. And I have to say, so Stuart's other book, Ignorance, really focuses on that. But even in the book Failure, this idea that, so the simple principle that blood circulates in the body, that we were probably 400 years too late in figuring that out because of that exact impulse of sort of, having this air of authority or expertise that was sort of so ingrained that no one would challenge the sort of company line, and that really slowed down scientific progress in an example like that. And so, proudly displaying ignorance, it turns out to be a really great trait for scientists to embrace.
All right. Hey, Nels, let's talk to Steve about some of the things his lab does.
I couldn't agree more. Let's do that.
All right.
The first thing on our list here is clam cancer. You just wrote a review article called A Sixth Modality of Infectious Disease. What are the other 5?
So let's see if we can reconstruct them. You guys will have to help me now, I'm
Fair
sure.
enough.
But there was a traditional sort of classification of infectious agents that we sat on and said, well, maybe this is really different. Maybe this is a 6th. And they were, I guess, let's see, viruses.
Right.
They were bacteria. They were fungi. They were prions. I think we thought it was another one was infectious. And the last one is, Nels, help me.
The 5th one?
5th one, rather.
Yeah, I'm just shuffling through the paper even as we speak.
Right.
Yeah,
Well,
so you got viruses, bacteria, fungi, parasites. Of course.
I guess so. It's kind of
Prions,
kind of a catch-all broad
Prions,
thing.
yeah.
Yeah.
Okay.
Yeah.
Those are,
Anyway, so he said this is different, and it's true. It's doesn't easily fit into any of those categories. And it was just kind of an interesting concept in general that cancers could be contagious, that they could actually spread from individual to individual.
Now, this was established with Tasmanian facial tumors, that they're contagious, right?
Right. So there really had been two examples in mammals before. There was the Tasmanian devil, and there was a dog tumor.
Dog tumor, right?
Right.
Venereal tumor of dogs, right?
Right. Exactly.
In these cases, the cancer cell is transmitted.
Right. So it's essentially a clone that established originally a normal tumor, and then the cells of that tumor developed the capability of spreading out to other individuals. So it's a kind of super metastasis where it's spreading. It's spreading not just within the body of a initial individual, but to new ones.
Now, in both cases, is the reason that the cells spread because there's some defective immune response?
Yeah, I mean, we try to think about what the parameters were that would be required to make this happen. I mean, you would first need a mechanism of transfer. In the case of the Tasmanian devil, it's thought to be through biting.
Right.
These animals do that. They're aggressive, and they do bite each other. So there's that mechanism exists. In the case of the dogs, it's thought to be transmitted venereally through sex. And then you need some mechanism for the tumor to be able to be accepted, in the recipient. And that would normally be blocked by an active immune system. So we think in both of those cases there would be probably some need for a defective immune response. Both of these are appreciated to be pretty deficient in MHC, but actually that's more common than you might think. I mean, a lot of tumors, even within a person, tend to lose MHC and survive attacks by the immune system in that way, even within one individual. But that may be part of the story. In the Tasmanian devils, it's thought to be also the case that the animals are so similar to each other genetically because of a bottleneck they went through in evolution that they might be almost unable to recognize the tumor as foreign. But it's not completely clear that that's true because the animals are divergent enough that they do reject skin grafts. So they do have some ability to recognize each other as foreign. But for whatever reason, they don't reject the tumor.
And is it true, Steve, so in the case of the Tasmanian devils, that transmissible cancer is really tough on them? It's uniformly fatal or close to fatal?
It is. It's pretty uniformly fatal. It's aggressive enough that there's hardly ever a survivor. That's different than the dog tumor, which is actually less aggressive, and the dogs quite often do survive, and they do reject the tumor. So
I mean, you can imagine in the case of where in the Tasmanian devils, because it is so fatal, that you could have this sort of strong then selective pressure if there's any individuals with resistance or escapers that would obviously be much more likely to then pass along offspring that themselves might be resistant as sort of an evolutionary counterpunch to this happening. And am I remembering correctly, I think something emerged in the last few months suggesting that might actually be happening.
That's right. So there are hints that that's the case, that there could be resistant animals. I mean, the selection is strong because it's so strong that they feared for the extinction of the species. So that's a pretty strong selective pressure.
Agree.
Interestingly, there's a second circulating tumor, so now known. So this tumor is rare, but it's not, given the right circumstances, it's not inconceivable that these things can happen more than once, and we'll come to that, I guess.
Yeah.
The dogs are not in one location only, right?
No, it's spread. I mean, that's a much older tumor. It's been around a long, long time, and it's spread all over the world. Okay. It's on every continent.
Okay.
Yeah.
So in that case, it's very different in the sense that it's not such a bad outcome. Not
right.
Yeah. And so has that spread then more widely? It can just sort of smolder in a sense in populations.
Yeah. I mean, it hasn't imposed such a strong pressure on the species.
And you're group got interested in another kind of transmissible cancer.
Indeed. I mean, we didn't expect it. We didn't plan for it. But some years ago, we started to look at a tumor that was known for a long time to occur in mollusks, in bivalves. The most characterized is in a species called the softshell clam, which I have an affinity for.
Yeah, a lot of us eat them, right?
Yep. Where I grew up near Cape Cod, it was popular. So, yeah, I was interested to learn from a friend, Carol Reinisch, at Woods Hole, that these animals had a significant incidence of a disease that's sort of like a leukemia, and she'd studied it her whole life, and she always, I think, had wondered if it might have a viral etiology, and I guess bit the bullet finally enough to call me up and say, listen, could we do some work and see, is it possible that there's a virus involved? So that was the start for us.
And
thought we would look.
These are clams that live in the wild, right?
Yes. They live in tidal flats in the mud, typically 6 inches to a foot into the mud.
And how did she figure that they had this leukemia-like disease?
Well, it's been noticed, I guess, that,
they die.
The heart, it's fatal, again, pretty invariably fatal. And the harvests suffer from that. And it was known to be kind of focal in the sense that a harbor, the most maybe notorious was New Bedford Harbor in Massachusetts, where the disease would appear and then spread widely and almost wipe out the harbor. And then they would, interestingly, from what we were saying, they often will come back after some years. So this hinted at something maybe infectious, although there was never any clear indication. A lot of times people attributed the focal aspects to pollution or some other exposure that might cause it. So anyway, we set out to ask maybe would there be a virus that was responsible, and we kind of did today what would be the usual ways of looking for that, which was to do sequencing, and we basically got RNA from the tumor cells in culture and directly from the hemolymph right out of the animals, and just did deep sequencing of the RNA with the help of Ian Lipkin, our friend across the way. We did a few other things to try to enrich for what might be a virus. We could sort of crudely purify virion particles if they existed from the hemolymph.
And can I ask, so how do you distinguish a cancer cell in a clam from a
what is
sort of a regular cell?
What does a sick clam look like? I guess you're asking. So the diagnosis is done by sticking a needle in and taking out a small volume of hemolymph, putting it on a microscope slide and looking at it. That's fortunately something you can do in the field, as it were, on a boat or in the mud, and look at it. And normal hemolymph cells from a healthy clam are essentially like macrophages, so they're adherent, they spread, they attach. These are the phagocytic cells that eat bacteria, keep the animals healthy. And the diseased animals instead have cells circulating that are very distinctively different. They're round, they don't attach, and the most amazing thing is the incredible hyperplasia. So these diseased animals have concentrations of cells that are just off scale. They're 10 to the 8th per mil, instead of 10 to the 6th or less per mil. So the hemolymph is milky with the cells. So you can a really sick, dying animal is it's incredibly obvious.
I see. Yeah. So and then the you started by sort of a really beautiful paper that came out. Was that already 2 years ago along those lines?
At least this was about reporting steamer, which was the retro element, as it turned out, that we discovered.
So it wasn't a virus you found, but
right, related. Yep. So, so in that RNA-seq data, where we were looking for anything that might be present in the tumors and not in a normal, what jumped out immediately was an enormous expansion of expression of an element that was homologous to our favorite virus, the mouse leukemia virus, and ultimately cloning and sequencing the whole element made it clear it was an LTR containing retro element, but not likely a retrovirus in that it had no envelope protein. And it was clearly a member of a well-known family that's present widely in nature, the Gypsy mag3 TY3 family of retroelements.
Yeah, very cool. But so what about sort of cause and effect? Which came first?
Well, no, that's still definitely up in the air. All we knew was that this thing was dramatically expressed in the tumors, and is it the cause? Don't know. But our next question immediately, of course, was to ask, okay, well, is there an insertion of this element, maybe that's in some crucial spot in the genome? And we still are working to answer that question. But immediately when we started to look at it, we ran into some surprises. And the first surprise was that there was an incredibly huge expansion of the DNA copies of this element in the tumors. Instead of the usual 2 or 5 or 10 copies in the genomes of healthy clams, there were about 150 copies.
Wow.
So that's one of the, I think, most dramatic expansions of any retroelement in nature. And if that had been happening in every clam every time they got cancer, that would have been pretty astonishing.
Agree.
Do you think that expansion occurs within the lifetime of one clam?
So what we discovered was now that the amazing fact, the real wake-up call, was the realization when we started sequencing the junctions, that is the sites of integration of these elements in the tumors, that the integrations were identical in the tumors from Long Island and Maine and Canada. All the tumors, in fact, had this exact same new integration sites. So they were not happening in each clam every time it got disease. It had happened in the past in a clone, and what we were seeing was now the dissemination of that clone.
Right.
Yeah, and so I think that raises sort of the next chapter, which was this absolutely beautiful paper that came this past summer on the widespread transmission of these cancer lineages, not just in that one species, but in many. And so is that what kind of led into this next?
Right. So as soon as we sorted out that first situation with the first species, the softshell clam, we got in contact with more marine biologists who were studying similar diseases in a number of other species. And some of those were in Vancouver, and some of them were in Spain, and they were in a variety of species. So we immediately said, wow, I wonder if this is happening in a similar way in all these other species. So we got samples, and again, we got the same story. In each case, there was a clone. It was a single clone, typically, that was spreading in a species, in that species of origin. In most cases, in one case, we had two clones that were circulating, not unlike the situation now with the Tasmanian devils. And then in one case, we had really the remarkable situation where the tumor had spread to a different species. So it was actually no longer found spreading in the species of origin, at least not commonly, but was now spreading commonly in another species. So it had jumped.
And in all of these cases, is a transposable element involved, a retro element, or not?
No, and in none of the new cases have we found any expansion of at least the guy that we know best, this guy Steamer. Of course, there are always many families, and maybe we will find another family that we don't know about that's expanded in these other tumors.
But you just looked at the clonality of the tumor in that case, which you can do by microsatellites, right?
Exactly. I mean, we can just look at nuclear DNA. We don't need to look at a particular retroelement for that.
One of the things that just seems really exciting about this to me is how this has sort of very quickly expanded into sort of this comparative, evolutionary approach that you take, in a sense, first of all, it has these echoes of some of the evolutionary biologists and field biologists I was hanging out this last week when we're now all of a sudden, I could imagine you booking tickets to Vancouver or Spain and getting out in the field and getting some mud on the boots, so to speak, which sounds really fun. But then that insight that it's not going to be the steamer element in every case, really fascinating. And then that curveball of that cross-species transmission these really kind of opens up how you can think about these things just by getting a few more points of comparison and then seeing that. It's really exciting to see how that's opened up in a really amazing way.
From the things we were talking about, what the requirements were for these things to appear, I guess there's the issue of how a tumor in a mollusk is transmitted, and we certainly don't know, but we do know the cells can survive in seawater pretty well. Certainly in the lab, you can put them in seawater and they're viable. So we guess they would have to be released somehow from a diseased animal, dying animal, and they'd have to survive in the seawater and then get picked up by a healthy animal. All of those things seemed possibly difficult, but in talking to the marine biologists, they were not at all worried. They pointed out that even healthy animals in this way can actually release quite a lot of hemolymph. They're in poop, they're in feces, so they're shed all the time. So they said, that's no problem, and they'll be in the water. And in fact, it's also not hard to imagine how they'd get picked up, because these are all incredible filter feeders. So these guys filter huge volumes of water, just liters.
Yeah.
So they easily could pick up a clone of a cell, and if it can establish itself and start the disease again, it's pretty easy to imagine how that could happen.
But the steamer-associated malignancy goes from this area, the Northeast, all the way down to Florida. Is that right?
Not that far south. It's Chesapeake Bay, and the softshell clam is doing well there, I might add.
It's still pretty far.
Pretty far. Yeah, no, it's certainly over, hundreds of kilometers.
But the clam people think that's not a big deal.
It would presumably be going from clam to clam to clam.
Yeah.
And they're only a foot apart.
Could be bilge
in the
water.
mud.
Could it be bilge water in boats too?
Yeah, and I mean, even the species has been transmitted to the West Coast probably by that route.
And am I remembering correctly? So in the case of that cross-species transmission of the contagious cancer, that actually the sort of donor species doesn't have a high prevalence of cancer when you just sample them compared to the recipient.
Right.
Yeah. And so what's going on with that?
So we don't know. But one of the points you raised might be happening. That is to say, they might have gotten resistant.
I see.
So maybe they're no longer susceptible. These two species coexist in the same beds. So there's certainly, they should both be getting exposed to it all the time. But only one of the species is now seemingly susceptible.
And are you sort of chasing after that possibility and actually thinking about what the sort of genetic mechanism could be for resistance in some of these cases?
So we'd love to try to develop these into real genetic systems. Obviously, these are not routinely studied genetic animals.
Yeah.
The genome of the softshell clam is just done and not annotated well. Mm-hmm. The only really well-studied genome is the oyster, which doesn't get this disease very commonly. So too bad for us. Yeah. So
you can't do anything in the lab. You can't transmit the tumor to another clam, right?
You can go from animal to animal. Yeah. Mm-hmm. Yeah. You can do it you can do it by injection, or you can even co-tank them, sort of the equivalent of co-caging mice.
Yeah.
And they will transmit from one animal to another.
So that brings the question, could you remove all the steamers from a cancerous clam and then transmit it and see if that's needed? Because that would ask whether steamer is needed or not, right?
Yeah, I guess it could.
It would be hard to do,
it would be very hard. I think, our view is and Michael Metzger, who's in charge here, is doing this. We're thinking if we simply sequence the genome of the cancer line and a healthy clam and compare them, we'll presumably be able to look at the 150 new insertion sites. And maybe we'll be lucky and one of them will be next to an obvious candidate for an oncogene.
Yeah, good idea.
Given, what's your thought? Do you think steamer is responsible or is just a bystander?
I think it's pretty likely because this is such a dramatic genetic, event that I think it's very likely the cause.
But in the other bivalves, there was no amplification.
There was not, or at least not. Well, no,
you didn't see any.
We didn't see any, but we're only probed, using the probes that we have for steamer.
Yeah, okay.
And that's certainly not surveying all the family of repetitive elements.
You didn't do the genome of those.
No.
Okay.
I have not done
so that would reveal it, right? Yeah.
it might.
So are we in the company of mollusks on this floor in the building?
We are. We are.
Oh, cool.
So we have a refrigerator that we set to 10 degrees, which is about what they like, and can maintain them. They're surprisingly easy to maintain.
Yeah.
They're pretty happy in the lab for months.
Sounds like a spectacularly fascinating system. And you mentioned Michael Metzger.
Did you know him, Nels?
I do. So he and I overlapped a bit at the Fred Hutch Cancer Research Center when I was a postdoc. Michael was a grad student with Dusty Miller before coming to Steve's lab. And you could already see sort of the glimmer in Michael's eye as a really great and talented scientist. And wow, this project, it just sounds like, to me, there's just so many possibilities as we're discussing moving forward. What an incredibly exciting system to develop as Michael continues his career.
Yep.
You think there are any transmissible human cancers?
Well, we've tried to think about that, and there are sort of examples. They're not exactly high frequency events. There are situations where there's maternal to fetal transmission. There are sort of transient cases, rare, where a surgeon who's working to cut out a tumor can inoculate himself, and the tumor is rejected typically because of a functioning immune system. Yeah. There's a really interesting case Nels will probably talk about where a supposed tumor turned out to be a parasite rather than a tumor. So again, it's kind of these transmissible tumors are kind of fuzzing the border between parasites and infectious agents and tumors. Yeah,
I agree. A new modality, a sixth modality of infectious disease. Yeah.
I guess you could take human tumors and look for evidence of clinality, right? People must have done that.
Yeah, one of the settings I thought where it would be plausible would be something like Kaposis sarcomas. Yeah. Of course, we know the virus normally transmits the tumor, but in that normal way, the virus is initiating a new tumor in the new infected person, and the tumor, of course, is his. It's not the donor's. But I still wonder if some subset of transmitted Kaposis tumors might, in fact, be examples of cellular transmission. So we are working to get the samples that will let us do that.
I can't help think of these horrible experiments done years ago with injecting people with Hela cells. You remember that?
Yeah, that was on my mind too. And even injecting is a pretty dramatic case, but even just folks working with Hela cells, right? And then those are like the dandelions of continuous cells.
That's right.
I think we all believe when you work with human cells that you're counting on your immune system to protect you. Of course. And the basis on that basis, it's often been said that you're not allowed to work with your own cells. And I think that's exactly the fear, that if you work with your own cells in the lab and you might transform them with a virus or do something interesting, that they would become dangerous to you uniquely because you would not reject them.
Yeah. So you have that sort of layer of protection where even if you're exposed, which is probably happening constantly, even as we're speaking, there's so many Hela cells in
Vince's lab
across the world being used for experiments. And so these exposures are happening constantly. And in fact, it's very rare that you see any result of this at all. And in some rare cases, maybe what starts to look like almost like a proto-tumor or a small growth, but at least to my knowledge, this hasn't gone on. And I think that sounds exactly right to me, that because we have immune surveillance in place to recognize these things as foreign, that acts as a really effective barrier in almost every case to block these things.
But many people are immunosuppressed severely. Well, all the AIDS infected people, right?
Yeah, HIV patients.
HIV. I wonder if some of the tumors are clonal there, right?
Well, in fact, so Kaposis is transmitted very much in that setting.
So you want to look at Kaposis tumors, right, to see if there's any evidence for that. Yeah,
right.
Yeah, that'd be interesting. It's a neat story. You didn't need any of the clams that you had in the lab, right?
No, although when we're looking for more examples of diseased clams, Michael, and we just go out to the stores, buy 100 or 200, and it's not uncommon to find one or two
really
that are leukemic.
Wow.
Yeah.
And you can tell
and
how.
they're in the market by bleeding them and,
You have to bleed them. You can't just tell by looking at them.
Nope.
But if a human,
Nope.
and I know reporters have asked you this, if you eat a diseased clam, it doesn't do anything because your immune system gets rid of it.
I wouldn't worry. No.
That's right.
Yeah, that's cool. The other thing your lab works on is actual retroviruses.
Yeah, that's certainly our main
Your main thing?
mainstream. Yeah.
This is a side project, and one of the things you've been very interested in which I know of because I've been on a number of your students' committees, is silencing of an integrated. Why don't you tell us the backstory?
Yeah, so I think one of the ways that was run into early on was situations where people were trying to use retroviral vectors to deliver genes to either hematopoietic stem cells or embryonic stem cells, and it proved extremely difficult. It failed. It was incredibly inefficient, and that was annoying if you were trying to do gene therapy and deliver a gene. So the studies showed that what was going on was that actually the virus was delivering the gene perfectly well, but after integration, it was simply silenced. It was transcriptionally shut off. So there were a lot of efforts to find promoters that would work, would not get shut off. There was some success. There were some efforts to find viral vectors that would not get shut off. And again, there were some success. There's in fact, a stem cell virus, so-called, because it was better able to infect stem cells. But it remained as a pretty interesting phenomenon that these were so potently silenced in these embryonic stem cells, unlike any differentiated cells where the virus was very well expressed. And a number of people worked on the mechanisms. Rudy Anish was one of the early players, Eric Barkless in his lab, a number of others. And what was worked out was that there were DNA sequences on the retroviral genome that were the target of the silencing machinery. It was realized that there was a machinery that you could detect in the nucleus of embryonic stem cells that would actually bind to those elements on the DNA and presumably mediate the silencing. Those binding proteins were not present in differentiated cells. And the situations kind of sat there for about 10 or 20 years. The technology really wasn't around to pursue it further.
Yeah, and so what was the key sort of advance that made it possible to get at the heart of this exact question?
So the key was really mass spec. So we were able to purify it partially in incredibly tiny amounts, but we had no way to characterize it until mass spec came along. And that's really powerful. It really allows you to work with incredibly, vanishingly small amounts of protein, get peptide sequence out of it, and because the genome is known and the sequence of all the peptides encoded are known, you can quickly identify the gene that was the source of almost any protein. It's really amazing to think that you can do that. You can work with very complex mixtures that are not at all pure and identify, dozens or even hundreds of the components based on the peptides that mass spec detects.
Yep. And what were the key proteins at play here that came out of?
So the first that we found was a protein called TRIM28 or CAP1. And that was satisfying because that's a well-studied protein that was known to be a silencer in a lot of other settings. So it's a widely used component of transcriptional silencing.
This was with a murine retrovirus initially, is that right?
So so these studies were all done in mouse cells, typically with viruses like our favorite, the Maloney murine leukemia virus, as the guy that was silenced. And it's worth saying the element where the silencing occurs is an interesting element. It's a short 18 base pair element in the proviral DNA, and it's interesting because it's the sequence that's used in the context of the viral RNA to initiate DNA synthesis, to initiate reverse transcription, and that initiation occurs with a primer, and the primer is a short RNA, tRNA, and of course, the retrovirus people here will know that all retroviruses and even retroelements initiate their DNA synthesis with one or another tRNA. So each element, each virus has evolved the sequence, this little primer binding site PBS sequence that is complementary to the 3' end of a particular tRNA. So the mouse virus that was so well silenced uses proline tRNA by chance.
So when you try to infect a mouse embryonic stem cell, the viral DNA integrates and then it's silenced.
Yep, exactly.
Now, the DNA also contains lots of endogenous retroviruses as well. They're all silenced as well?
Indeed. So, in fact, we would guess that maybe it's really the silencing of those endogenous elements that's the basis, the evolutionary pressure for the development of this silencing system. It's probably way more important to keep those endogenous elements silent than the occasional exogenous virus that might wander into these cells.
Yep.
Well, the original exogenous infection, before there were any endogenous elements, would have killed the stem cells if it hadn't been silenced. Maybe the one that survived was silenced, and all the mice are derived from that one.
Fair. Yeah.
So, well, certainly for evolutionary virologists, the idea that sort of the expansion of the TRiM family of proteins and even the evolution of specific orthologs or representatives of members of the TRiM family are under rapid evolution has been sort of a long known. And so that sort of fits from an evolutionary standpoint with exactly the idea that you're saying, that you have these potential for recurrent genetic conflicts where you're selecting on variants that might enforce that silencing, whether it's a exogenous, an endogenous, or a retro element. And then that sort of is a source for diversity where one of these things might be useful or beneficial given ongoing or a new infection.
Right. So the TRiMs are clearly a family of tools in our toolbox that have evolved to restrict or silence retroviruses. TRiM28 is an unusual member of the family acting at this level of transcription. Most of the other TRiMs, we think, are antiviral at other steps, TRiM5 being the one that's most characterized, but others, TRiM25 being characterized. And these often have broad antiviral activities for flu and other viruses as well.
So and it's not just the TRiM proteins that you pulled out.
So TRiM was interesting and good for us, but it clearly was not the whole story because TRiM28, in fact, itself is not a DNA binding protein. So that wasn't the guy that was recognizing the proline PBS. And it also wasn't at all embryonic stem cell specific. As I say, it's widely used. It's constitutive. It's expressed everywhere, and it's widely used. And it's tethered to its target genes most often by one or another of an interesting large family of proteins, the zinc finger proteins that contain a crab domain, which is the site of interaction with TRiM28. So these zinc finger proteins are very interesting also, evolutionarily.
I agree. So another one of these hot spots for rapid evolution. And in fact, among some of my colleagues, in essence, if you start to try to tease apart some of using a comparative approach or a phylogenetic approach to look at some of these families of zinc finger proteins, it can feel like you're flying a little too close to the sun because there's so much rapid evolution going on that it becomes really tricky to sort of make robust comparisons. There's just so much volatility and change. And so I think that really highlights how these things could be right at the center of these kind of silencing mechanisms.
So when we purified this DNA binding complex further, along with TRiM28, what we found was one of these zinc finger proteins. It was a particular mouse one called CFP809, and it was not previously known, but it turned out to be, in fact, the exquisitely specific DNA binding protein that was recognizing the proline PBS. So it's very clear this guy evolved to bind proline PBS and silence retroviruses. And it's very easy to imagine, I think, how lots of these members of the family could evolve to target whatever is needed.
So if you knock down this Z 809, what happens to the cell?
So Todd McFarlane at the NIH has done that. He's actually made the knockout mouse. And what happens is, yes, all these many endogenous guys come on and are not properly silenced. And it's very bad for the mouse to have these turned on.
Is it embryonic lethal?
I think it's lethal at some point, yep.
Now, if you induce in ES cells to differentiate. At some point, do these silencers go away and the genes are turned on?
Yep.
Yeah.
So partly true. That is, the complex goes away, the DNA binding activity goes away, but typically they don't come on. And the assumption is that they've been silenced, and the silencing is maintained in a way that no longer requires the continued presence of the protein. And one of the ways that happens is through probably DNA methylation.
Okay.
So once a certain stage is reached, they're off, and it takes hard work to get them to reactivate.
So an adult mouse doesn't have much expression of these endogenous loci, basically.
Right. And they don't typically also have much expression of 809. So they're
some other mechanism.
They've gotten past the stage of danger, I guess, where the suppressing is needed.
Now, in some cases, we know that, at least in humans, you need some retroviral expression during very early development, right, to maintain pluripotency, for example. And so are there similar observations in mice?
Yeah, so I think we believe that many of the endogenous elements are not bad, but are doing good things. They've been co-opted to do good things. They're such a huge part of the genome. They represent transcriptional promoters that are handy. They're often driving genes that are important. And, it's not the case that they're all simply silenced at all, period. Some come on, some are off. They all have complicated temporal profiles of expression. And I think the retrovirus promoters are simply part of evolution at this point of all our genomes.
Yeah, and it's a good tie into your science paper that we talked about on TWiEVO last year, right?
Yeah, that's right. Things as you sort of dig beneath the surface. So we found this connection between this enrichment for these motifs that are sort of donated in a sense from these endogenous retroviruses, these LTR elements that have binding sites for so-called gamma-activated sites to attract the transcription factors that help to underlie an interferon response where you upregulate hundreds of genes. So it's as if these ancient retroviruses that were potentially sort of listening in on our own immune signaling then came online as they're endogenized in our genomes, and then as they dispersed, some of these elements were reclaimed for us to fight viruses today. And so you have these sort of crazy connections that can sort of unfold both for the better or the worse, sort of.
Right.
For better or for worse, in sickness and in health, we're with the retroviruses and they're with us.
But the viral genomes, even if expressed, they don't make infectious particles, right?
Right. I mean, the vast majority of them in mice, and not all, but the vast majority are defective. And as far as we know, all of the HERVs, the human ones in us, are defective. And it's presumably a function of their age. So the older the families are, the more likely they will have all gone defective. And I think, our concept now of how these things are appearing in the genome is that there are relatively short periods of time when expansion occurs. So that may have happened, tens of thousands of years ago, hundreds of thousands of years ago, or millions of years ago. There would have been a dispersion. There would have been an epidemic, if you will, and they entered the genome and dispersed, and then they've gone defective since then. So if they're very old, they're very defective. The most recent we know of in humans is the HERV-K family, and they're
a million years ago.
Yeah, well, and they're maybe younger than that a little, and they're relatively close to being functional, such that it's been possible to, in fact, correct the mistakes that we can see and regenerate functioning viruses.
That's why the koala story is so cool, because we can see that happening now. Because the interesting question is this happening to humans somewhere? We don't see it, but we see koalas being endogenized, right?
Yeah. There's also, I think, another case of deer herds up in Wyoming that have been proposed to have active elements. And so this, again, we have the same echoing from our first half of the show, thinking about these clams in Vancouver and in Spain, and now you can chase around koalas and deer if you want to get some comparative ideas for this kind of biology.
Plenty of opportunities for field work.
Yeah, this is an interesting collision between virology or cancer biology and field biology as you start to put it into this evolutionary context.
Now, you've done also some work with HIV silencing, right?
Yes, and the context where people are most interested in HIV silencing is in the latency issue.
Mm-hmm, right.
So, I mean, one of the most important problems in HIV today is the fact that no matter how effective your antiviral treatment is, there's always a latent reservoir, and that's going to be very difficult to get rid of.
Right.
But people would love to know more about how those genomes in the reservoir are kept silent.
Yeah. It's not an interest in germline because HIV doesn't infect the germline.
Right. Yeah. These are just memory T cells, and they're very long-lived, decades. And so they lie silent. The virus is silent. It's very hard to know how one could identify the cells, let alone get rid of them.
So is it simply a matter of finding a key protein like TRIM28 and silencing it?
Well,
then blast them with antivirals, right?
Silencing a silencer.
Yeah.
Yeah.
Yeah. I mean, it's a serious effort a lot of people are trying to make. Yeah.
But TRIM28 is not involved in HIV silence, right?
It doesn't look like there. It could be. I mean, I think it is possibly one player. Yeah. We don't know of any zinc finger that's specific for HIV. There's a bunch of other silencing proteins that have been suggested, and it may be that there are layers of multiple layers of silencing.
Okay. In fact, you mentioned the idea of methylation as a post-developmental silent. In fact, you just published a paper about that, right, from your laboratory, which describes that, I think. We'll put that in the show notes.
Yeah, good idea. And I don't remember all the details, but maybe you can help fill in some gaps here, Steve. So there are now, I think, even in maybe early-stage clinical trials, some pharmaceutical compounds that actually are affecting things like chromatin structure through some of these marks that are proposed to actually kind of wake up these latent.
Yeah. People are trying to use SAHA and other drugs that would maybe change the chromatin state and activate. And I think they'll work at some level. I mean, I think you can induce HIV inefficiently out of these cells. And I think the challenge that I don't see an easy way is to get them all. I don't see any easy path to literally getting every cell.
Yeah, I completely agree.
You just need one, right?
You probably need very few, and the infection will be reawakened.
Yeah, it's a real problem because nothing is 100%, right? Yeah.
Nothing's 100%. When someone says your antibacterial soap is 99.9% effective, right. It's that 0.1 you might want to keep an eye on.
Yeah, it's always a problem. Are you okay on time, Steve?
I'm good.
All right. I want to tell listeners about sponsor this episode, Blue Apron. Their mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting high standards for ingredients, and building a community of home chefs. You know, scientists like to cook. Do you like to cook, Nels?
I do, yeah. So, as you sort of go through the stages of your career of spending a lot of time in the laboratory you working with your hands to produce something, to run an experiment, and then that sort of slips through your fingers and you spend a lot of time in front of a computer. It can be really therapeutic to get into the kitchen. And
it's a good point. We're moving away from wet science and doing more computational stuff.
That's right. And this is sort of a way, at least for me, to reconnect with going through a protocol, a recipe, and working with my hands, putting something together. And, what's really nice about a cooked meal is you have the product. It works. Well, not every time, but most of the time. Whereas I would say it might be the inverse for the experiments we conduct, where it's that rare day when something comes through. So cooking is, I think there's definitely a sort of kindred spirit here with the scientific
I totally agree. I love to cook. I'm the cook in the family. You bet.
Yeah, that's why I thought Blue Apron would be a good interest for the fraction of our listeners who are scientists.
You know what it totally reminds me of, which I love, is it sounds like Lake Wobegon, which has great ads.
Really?
Oh,
really?
Yeah.
Well, what Blue Apron does is they deliver seasonal recipes along with everything you need to make a meal. Everything but salt, pepper, and oil, basically. Every meal comes with a step-by-step recipe card with beautiful pictures, and it's basically in 40 minutes or less you're done. And it's very inexpensive. If you spend a lot eating out at high-end grocery at restaurants or high-end grocery chains, you can spend less than $10 a person for a home-cooked meal, which is pretty cheap, I think. And they get their food from a network of farms, fisheries, and ranchers across the U.S. They have 150 different people that they deal with. For example, seafood they source it sustainably using standards developed together with the Monterey Bay Aquarium Seafood Watch. They get their meat, beef, chicken, and pork from responsibly raised animals, produced from farms that practice regenerative farming. And they give you exactly what you need, so there's no waste, and you can't overeat. That's the good part, too. Unless you cook too many portions, of course, I guess you could do that. You can tell them when you want deliveries. There's no obligation. You can tell them meat or no meat. They deliver to 99% of the U.S., and they don't repeat their recipes in a year, so you never get bored. So this month we have salmon piccata with orzo and broccoli. We have pork chops and miso butter with bok choy and marinated apple, spicy shrimp coconut curry with cabbage and rice. Those are just some of the examples of what you can get.
And we need to get them to do clams.
Right, clam chowder.
Yeah.
Clams. Check out this week's menu and get 3 meals free with your first purchase with free shipping. You go to blueapron.com/TWiE, TWiE. That's blueapron.com/TWiE. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron. So don't wait. Blue Apron, a better way to cook. I really like it. I've tried it.
I've been on the road too much, but
you should try it.
Again, my mouth is watering. I'm thinking about a second lunch after that. And I also have to say, I like Steve's idea of hearkening back to the Lake Wobegon days. If we can, maybe we should see if we can get Powder Milk Biscuits on as a sponsor down the road.
Exactly.
Heavens, they're tasty and expeditious.
I never listened to it.
Oh, you're missing something.
I grew up in Minnesota, so I was.
It
was not an option. It was mandatory.
Nels, should we do a couple of picks and wrap it up?
Yeah, let's do. So I wanted to return a little bit to this theme of microbial mind control or parasitic control of behaviors. Yeah. And so I'm going to try to position to show you guys a couple of really striking photographs. So this all comes from a National Geographic article entitled Mind Suckers that was written expertly as usual by Carl Zimmer. This is the November 2014 issue, and we'll put in the link. And more than that, there is a fellow, Avind Varma, who is a really good photographer of sort of biological scenes. And so he did just this striking collection of photos of, in many cases, insects, but other critters being manipulated actively by parasites and microbes. So we'll have this up on the website as links, but I'm showing you guys a picture of a cricket that was, crickets usually don't swim; they stay dry. But this one has jumped into a puddle of water.
That's right.
It's a little bit staged for this photo, and what's happened, and this is part of the life cycle of a parasitic worm, the horsehair worm. That once the cricket is wet and drowning, it emerges from the cricket, swims out, and then goes on to infect other crickets. So that's one. Here is a spider that is giving a piggyback ride to the larva of a parasitic wasp. I don't know if you can see that little thing there.
That's great.
Yep.
Yeah.
And so, the manipulation, the basis of this isn't known. Is there a virus or another microbe at play? But what happens is the spider will actually stop spinning a proper web. It will spin this really modified strange web that, as the larva is developing, can use as a cocoon. So basically, the larva is snacking off the juices of the spider while it's building part of its cocoon for it, really controlling the spider. Here's one that you might like, Steve. We're back into the seawater here. This is the sheep crab that's being parasitized by these parasitic barnacles. And what's happening here is that the male crabs actually get feminized. So their morphology changes. This provides sort of the proper location that's perfect for the barnacles to sort of set up shop and reproduce. And then the claws of the crab or some of the appendages change form. And then the behavior of this is to just sweep the seawater over the barnacles that sort of, again, facilitates its reproduction and its dissemination. anyway, I could keep going and going. We've got fungus zombified ants. We've got nematodes infecting and changing the abdomen color of other ants. We have amphipods infected by worms that are all of a sudden swimming to the surface of the water right when they shouldn't, to be consumed, again, causing this sort of replication. So anyway, all this just gorgeous photography and biology playing out as microbes and parasites are manipulating their hosts and changing their behavior in really crazy ways.
That's very cool. Yeah, we've talked about a few of those on TWiP.
Oh, yeah.
This is the cricket story that the parasite makes them jump in the water. And I think they get eaten by fish and it spreads the worm, something like that.
Yeah, exactly.
Right.
And so is it that the parasite, something the parasite's doing, or is it deploying a microbe in some of these cases? And that remains an open and interesting question, I think.
Yeah.
So they're sort of symbionts, but they're sort of exploitive symbionts. They're not always to the benefit of both. Some of it's one way.
That's right.
Yeah.
So this idea of conflict versus cooperation, are these sort of two sides of the same coin, or how do these things kind of break in different contexts? Yeah, it's
cool stuff.
Great question.
Yeah, that's a neat one. All right, my pick is a little YouTube video over at Quanta Magazine, which Steve knows well. It's put out by the Simons Foundation, where your wife works, right?
Indeed.
And I spent some time being on the advisory board for this magazine. They have a series of podcasts and videos, and the videos, which they're trying to do more of, are pretty well done. And they have some experts explaining various things. They're very interested in math and physics and that sort of thing. But they have one here which I like. It's called "What is a Species?" Now, there are lots of... You search for that on YouTube, you'll find hundreds of videos where everyone tries to explain what a species is. You just don't have Charles Darwin doing it because he wasn't around for videos. But this is David Kaplan. He explains why a simple definition of species is hard to come by. So this is one of a series of theory videos. It is, 2.5 minutes long, which is perfect.
Yep.
You deliver a message, and they're visually very nice and engaging. And so I think it's worth our listeners to check it out to see why it's so hard to define a species, especially when you talk about viruses, right? I mean, the original definition is a species doesn't mate with another species and produce viable offspring, right?
Yep.
But with viruses, it doesn't make any sense. There's no mating.
That's right. That's right. So, and if you go back to, or if you watch that video and it sort of, gets you curious about this, and you could go back to TWiEVO episode 4. Mm-hmm. So this idea of division of species, goes all the way back to Charles Darwin's day, what he called the mystery of mysteries. And so we had a science buddy of mine and colleague, Nitin Fudness, who works on this exact question in fruit flies and Drosophila, hybrid viability and fertility things.
Yep.
And so he gives us a play-by-play of sort of the state of the art for some of these topics.
There's just gray zones. There's just, areas where sometimes only in one pairing, male-female, can go and hybridize, and not others. So
that's right.
Species are fuzzy.
Species are fuzzy, and if in the more where you can start to get a genetic foothold on some of how that fuzziness is defined, it's really teaching us a lot about very basic fundamental questions in our own biology, but also even some repercussions about thinking about fertility and reproductive biology as well, that certainly a lot of folks care a lot about.
Think about humans and Neanderthals, right? Hey.
Yep.
Think of them as different species, but at some point they weren't.
That's right. And we've spoken about that on TWiEVO as well. So this idea that there's sort of these little echoes of Neanderthal sequence integressed into our own genomes, with potentially some consequences for some of our immune functions that in essence might have been provided by some of those archaic species close to us.
Provided.
And Neanderthals have endogenous retroviruses.
They do, yeah. Everybody does, right?
Yeah. Yep.
About tetrahymena?
Oh, yeah. So there's quite a, even there things get even more wild, which is not unusual in the ciliates. So the division of nuclear labor, there's a germline in a somatic genome. The germline genome is chocked full of repetitive elements that are then trimmed out through these sort of modified small RNA mechanisms And then in the development of the somatic or so-called macronucleus, these things are all trimmed away, and you're left with these really gene-rich microchromosomes that are amplified up. And then those are the ones that are where transcription and translation of proteins are happening. And so this might be basically the because there's so many of these elements that this has caused this really sort of complexity to emerge in nuclear morphology.
Our colleague Laura Landweber works on that. You should have her on board.
Yeah, great idea.
She'd have a great time.
Yep.
Yeah, we should bring her on to TWiEVO sometime. Right?
Agreed.
Well, that's TWiEVO 17. You can find it at iTunes, microbe.tv/TWiEVO. And you can send us questions and comments to TWiEVO@microbe.tv. And if you'd like to help us out financially, support our science shows, go to microbe.tv/contribute. There are a number of ways we have for you, including Patreon accounts where you can subscribe and help us out every month. Our guest today has been HMI investigator, professor of biochemistry here at Columbia, Steve Goff. Thanks for coming by.
It's always a pleasure.
I know you came so far. I really appreciate it.
Right.
Nels Elde, you can find him at cellvolution.org, also on Twitter as Earlybird, usually at University of Utah in Salt Lake, but here in New York City today. Thanks, Nels.
Yeah, I came from a little bit farther, and I have to say it was 100% worth it. It's really a fun honor to be in the presence of you two.
It's fun to do it in person, you know.
Agreed.
We should do it more often, but
it's definitely fun.
Distance.
Yeah.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious.